Actively dividing cells pass through a series of steps known as the cell cycle. There are two main phases in this cycle. First, there is interphase, that portion of the cell cycle in which the cell simultaneously carries out its work and, in preparation for division, duplicates its chromosomes. Second, there is the mitotic phase, or M phase, that portion of the cell cycle that includes both mitosis and cytokinesis. The cycle begins when cells, recently created by the division of parent cells, grow, duplicate organelles, and prepare the machinery needed to replicate DNA. This is called the G1 phase, and it signifies the first gap between the birth of the new cell and DNA replication. The next stage of the cell cycle is the S phase, or synthesis phase. During this stage, each DNA molecule in the nucleus is duplicated. When DNA replication is complete, the cell enters another gap stage called the G2 phase. Cell growth and the duplication of organelles continue as the machinery for cell division is prepared. Together, the two gap phases and the S phase make up interphase, an interval during which cells do not divide. Cells spend most of their time in interphase. The cell then proceeds through a series of events, collectively called M phase for mitosis, that result in the formation of two daughter cells. In preparation for cell division, chromosomes must be duplicated and then distributed so that each daughter cell receives an identical copy of the complete genome. During the G1 phase, cells possess the standard diploid number of chromosomes, in this example, 4. Each chromosome contains one DNA molecule. During the S phase, each DNA molecule is copied or replicated, and the two sister chromatids remain joined. Thus, the number of DNA molecules doubles to 8 during the S phase, but the number of chromosomes is still 4. During the G2 phase, both the number of chromosomes and the number of DNA molecules remain constant. In M phase, mitosis, the nuclear membrane breaks down, and chromosomes become separated into two equivalent sets. The chromosomes condense, line up in a row, and then the sister chromatids separate and move to opposite poles of the cell. After the chromosomes separate, the nuclear membrane reforms around each set of chromosomes, and the cell undergoes cytokinesis, the division of the cytoplasm. Finally, when cell division is complete, each daughter cell contains a complete set of chromosomes. That is, each daughter cell has 4 chromosomes and 4 DNA molecules, as did the parent cell. How is the cell cycle controlled? Normal cells halt their progress towards cell division at a series of 3 checkpoints, which occur during G1, G2, and metaphase. At these checkpoints, proteins survey the cell to ensure that conditions for a favorable cellular division have been met. At the G1 checkpoint, proteins in the cell determine whether cell division is called for. To do this, the proteins survey the cell's environment for the presence of other proteins called growth factors, which stimulate cell division. When growth factors are limited in number, cell division does not occur. If enough growth factors are present to trigger cell division, then other proteins check to see if the cell is large enough and if sufficient nutrients are available for the cell to synthesize its DNA and divide. At the G2 checkpoint, proteins ensure that the DNA has replicated correctly and again make sure that the cell is large enough to divide. The third and final checkpoint occurs during Metaphase of Mitosis. Proteins present at metaphase verify that all the chromosomes have attached to the microtubules of the spindle so that cell division can proceed properly. If the systems surveying the cell at any of these 3 checkpoints determine that conditions for cell division are not met, the progression towards division is halted. When this happens, the cell may die.